fantastic to be here. I want to thank Jane and her team for pulling this together. A very important subject. I sure am learning a lot about some of the classic approaches in toxicology. I've entered into this field from a different direction, worrying about endocrine disruption. And what Jane has asked me to do today is, because many of you will not be familiar with some of the basics of that field, I'll quickly summarize that and then ask questions about how do key elements of endocrine disruption challenge some of the assumptions that underlie toxicology generally and TDCs in particular. So why worry about EDCs? Well, the World Health Organization this year issued a major report concluding that endocrine disruption is a global public health threat. World Health Organization. And then, again, about a year ago, the CRO Forum, you're all Swiss, you know what the CRO Forum is, the Chief Risk Officers Forum of places like Swiss Re. They issued a report on EDCs, basically saying that the highest risk for the insurance industry. So here the reinsurance industry insures the insurance industry against disasters overcoming companies that the insurance industry is insuring. Okay, so the reinsurance industry pays attention to big financial risks created by manufacturing and use activities. And they concluded that the highest risk for the insurance industry in this issue emerges from the probable link between low-level EDC exposure and bodily injury. This came from Swiss Re, based here in Zurich. They recommended that the companies that are insured by the insurance companies that the reinsurance industry insures, I know it's a complicated trophic system, but that's the way it works globally, that they recommended that those companies using EDCs or releasing them into the environment begin to lower those activities because of the financial risks associated with them, which can be very substantial. So why did the World Health Organization and why did the reinsurance industry reach striking conclusions like that? It's because if you look at the literature that's emerged over the last 20 years having to do with associations between specific endocrine disrupting compounds and specific human health endpoints, they are associated with varying degrees of certainty, some more, some less, with a wide array of today's hormonally related epidemics. None of these is it certain. As science goes on, some of these will drop off the list, others will be added, but I think for the most part, the science is going to become ever more certain for most of them. So what is an endocrine disrupting compound? According to the Endocrine Society, which is a professional association of physicians who help you recover from endocrine-related diseases, and scientists, research scientists who perform research on endocrine disrupting compounds and other aspects of endocrinology, an EDC is an exogenous chemical or a mixture of exogenous chemicals that interferes with any aspect of hormone action. 70% of the Endocrine Society is physicians. In my country, at least 70% of physicians are Republicans. This is not a fringe organization reaching an outlandish conclusion. They take this issue very seriously. It's the issue, the public health issue in which the Endocrine Society is most actively involved. So what did the WHO report conclude? First, many endocrine-related disorders are on the rise. It's happening far too rapidly to be the result of evolution. That takes generations. This is happening within 1 or 2 generations. There are 800 chemicals so far on lists that have been compiled that are in common use that are known to disrupt some aspect of endocrine function. We know that exposure is ubiquitous because of the global transport of endocrine disrupting compounds. There's virtually no organism on the planet that isn't carrying at least some body burden of endocrine disruptors. Over the last 20 years, easily over 10,000 studies have been published in the peer-reviewed literature, looking at laboratory experiments with rodents, looking at wildlife experiments in the lab, looking at people like Mikel Porta doing really incredible work looking at epidemiological studies. And in essence, they paint a consistent pattern from laboratory studies to wildlife studies to epi, saying that there are endocrine disruption impacts on human health. The WHO concluded that because there are so many uncertainties, for example, of the many more compounds, more than 8, of the many compounds that are in circulation in common use today, most of them have not been tested for endocrine disrupting endpoints. So there's a lot we don't know about that. There are a lot of endocrine systems that have not yet been challenged with tests, and the one thing we're pretty sure is that virtually every element of the endocrine system is at least theoretically vulnerable to endocrine disrupting compounds. Now, I think the most exciting part of this science is that this large body of literature is saying that there are really important opportunities for reducing disease burdens by getting the science and the regulation right. That's the exciting part of this picture. Now I can't deal with 10 to 20,000 individual papers for you, so I'm going to rise above that and extract a few central themes very quickly for what I think are the core elements of discovery in endocrine disruption. One is that EDCs interfere with hormone action, including through epigenetic alteration of gene expression. And increasingly, that's where the focus of this research is. These effects occur at extremely low levels. Level of effectiveness varies from compound to compound, but some of them are in the parts per billion, even low parts, or even beneath a part per trillion, at least in cell systems. Low-dose effects in the womb can have consequences that last the lifetime of the individual, where traditional toxicology looking at the animal or organism or person at birth will show nothing, but the consequences by my age, your age, can be extraordinary, simply because of fetal exposures, which are no longer measurable in adulthood because they took place in the womb. The exposures, as I mentioned, are ubiquitous. As we just heard a really great talk from Thomas about how mixtures can interact in very unexpected ways. Some you can predict, some we're really reaching to try and figure out how to deal with them. Here's a new dimension to this field, which was mentioned briefly in the discussion this morning. Some of these epigenetic changes that are induced by fetal exposures can be inherited not through changes in DNA sequence, but through changes in the epigenetic control of gene expression. And this is an oversimplification, but basically, great-grandmother is exposed while she's pregnant. The first generation shows the direct consequences of exposure. The second generation shows the direct consequences of exposure because that second generation fetus is forming, when the first generation fetus is pregnant, she's forming the eggs of her children in the womb during those exposures. So you've got you got mom, first generation, second generation, directly exposed. But these studies now carry it out to the next generation, the great-grandchildren, and those mechanisms clearly are epigenetically mediated. And a wide array of health endpoints have been shown to be carried out transgenerationally. There will be a study coming out probably next week that looks at a four-generation epigenetic transgenerational inheritance due to DDT exposure, for example. The tools that we have used to tell us what is safe and what is not for conventional toxic materials have some core assumptions that just don't work with endocrine disruption. They've got what we're calling neotoxic characteristics, and I'll be diving into them momentarily. As the WHO summarized, EDCs are now linked to a wide array of human diseases and disabilities that are increasing. You'll hear public health authorities say that this is the first generation of children since the Black Death who are likely to enter middle age less healthy than their parents, and that's particularly because of metabolic disorders, which Angel Nadal works on. For me, another exciting part of this is that we are getting really quite interesting design signals from our molecular genetic understanding of the direct mechanisms of action that are going to help chemists design new materials. That as this science advances, we'll be able to move towards a new generation of inherently safer materials. So those are the 8 broad points What I'd like to do is dive quickly into two. One is that, actually three. One is that these interactions can take place at very low levels. This is work on the endocrine disrupting herbicide called atrazine, which affects how rapidly estrogen is converted, or excuse me, testosterone is converted into estrogen through activation of excuse me, thank you, Anna. Through activation of aromatase, work by Tyrone Hayes at University of California, Berkeley. These animals were exposed from hatching to metamorphosis to adults at 2.5 parts per billion in the water. They're mating. The effect is visible in front of you, although you can't see it. The animal on the bottom is a fully functional female, but it's actually a genetic male. 10% of animals exposed in this strain of Xenopus are fully converted from genetic male that without exposure would have grown up to be a functional male. They're converted to being a truly functional female. They can lay eggs that are fertilized. Those eggs can grow up and become, again, genetic males, but functional females at 2.5 parts per billion. If you look in the environment, what sort of exposure does 2.5 billion parts per billion translate to in terms of environmental exposures? A farmer wants to have that's a log scale. A farmer wants to have atrazine on the weeds above 1 million parts per billion. That leads to a variety of levels of contamination in the environment downstream, ranging down to 80 parts per billion in surface water away from the stream of water flowing off the fields. Because atrazine volatilizes, it gets into the atmosphere, condenses in water in clouds, you'll find rain anywhere with about up to 600 miles downwind of atrazine application sites at 1 part per billion. EPA in the US considers about 100 parts per billion to be safe for human consumption over short term, and at 3 parts per billion is considered safe and long term. One of the motivating factors for the Chief Risk Officers Forum to release that report was that when water utilities in the Midwest were forced to clean up the drinking water of people living in those towns, the water utilities ultimately, because they couldn't meet the EPA standards, the water utilities ultimately went to Syngenta and sued them, and Syngenta settled for $105 million. Tyrone Hayes published his initial results about 8 years ago, showing that a partial conversion to female was inducible by 0.1 parts per billion, and the result that I just summarized for you about full conversion takes place at 2.5 parts per billion. So you can see the range of exposure levels sufficient to cause significant changes are well within the range of environmental experience. Second, let's look at this issue of low-dose exposures in the womb can set in motion things that play out over a lifetime. And that's the inside of a mouse. That's a normal mouse. It's a mouse my age in mouse years, getting on there. This is work out of University of Missouri. Note the kidneys are healthy, and the bladder looks like any mouse my age bladder would look. What would happen if that animal had been exposed during fetal life to 20 parts per billion of bisphenol A, and then allowed to grow up to my age in mouse years? The kidneys are necrotic. They don't work anymore. They haven't for some time. The bladder is about to explode. If the animal had been allowed to live, its bladder would have exploded in the next few days. And what's happened? There's a piece of tissue in the urethra, which is what we all pee through, that responds epigenetically to bisphenol A exposure in the womb. Those epigenetic triggers are changed. They then react through the lifetime of that individual differently to changes in testosterone and estrogen levels, which happens, all men undergo changes in testosterone and estrogen ratios. And the response that that section of urethra has is it gradually constricts because of those epigenetic changes until the animal can no longer pee, leading to an exploding bladder. Lastly, so what is it about endocrine disruption that, that challenges some of the traditional assumptions in toxicology, and I think therefore has some challenges for TDC? There's some false assumptions underlying the application of traditional regulatory toxicology. That list of epidemics, virtually none of the standardized assays that are used in toxicological testing are really relevant to the list of epidemics that I began with. Uterotrophic assay has nothing to do with any of those except the grossest indication that a chemical may be an estrogen. It tells you nothing about the sorts of epigenetic mechanisms that underlie the types of changes and the risks that we're talking about here. Second, it assumes that gavage is relevant to human oral exposure. Mouse experiments done with gavage, it assumes that those are relevant to human oral exposure. And think about that. That's a false assumption. And think of how many of those TDC data are established based on gavage experiments. Third, another false assumption is that high-dose testing, which is used throughout regulatory toxicology, assumes that high-dose testing can predict low-dose results that are caused by those epigenetic changes. So I don't want to spend a lot of time here, but the agency risk assessment that we all have depended upon don't incorporate modern molecular biology. It's sort of like, imagine move into a different field for a sec. Okay, think about, how many of you seen the amazing photographs of galaxies taken from the Hubble Space Telescope? Okay, those are amazing photographs. Imagine you want to prove that they don't happen. Go into your backyard with binoculars. That's an analogy with how old, outdated assays used today in toxicology do not work with modern molecular genetics. They're decades out of date. So here's gavage, used in probably hundreds of thousands of experiments that form the basis of a lot of the numbers that TDC depends upon right now. Gavage bypasses one of the main pathways of absorption of chemicals in people, which is the oral mucosa. It goes straight into the stomach, where it compounds used in gavage experiments go straight into the stomach. They're absorbed in the stomach. They go straight into the liver. They undergo first-pass metabolism, and a lot of them get out immediately through through peeing. If you were to allow the animal to actually put it into its mouth and have it absorbed sublingually, the pharmacokinetic characteristics of that exposure are extraordinarily different. They're faster, there's more, and there's no first-pass metabolism. That's a big problem for all of toxicology, not just EDCs, but just think about this for a minute. How do you take nitroglycerin if you've just had a heart attack? You stick it under your tongue, right? That's sublingual absorption. It turns out it's quite common in hormonally active materials. So all the experiments that have developed pharmacokinetics based on gavage are misguided, including some of the classic ones where you take a hard gelatinous capsule, you stick your compound in that and use that to deliver it to people, completely bypasses sublingual absorption. There are also stress issues. Gavage actually turns out to be a stressful incident for mice. And there are experiments now, lots of them, showing that a stressed mouse can be shifted towards the experimental mouse in the endpoints of interest and make it much harder to detect significant differences between your control and your experimental. So there are at least two major reasons why gavage-based experiments are very misleading. Lastly, the whole issue of high-dose testing not being sufficient to anticipate low-dose results is captured by this slide. These are mice experiments done by the National Institute of Health in the US, allowed to grow up to adulthood. It's the same strain of mice. no statistical difference in caloric intake, no statistical difference in the activity levels. But clearly, the experimentals, which are on your right, are clearly morbidly obese. What happened? These animals were exposed to 1 part per billion of DES right at birth. DES is now what's called an obesogen, in addition to all the other things that DES does. And a lot of obesogenic activity appears to be mediated by these obesogens hit PPAR gamma in mesenchymal stem cells, and those mesenchymal stem cells, which would have become bone cells, become fat cells. We're now learning that there are actually quite a few obesogens, not just DES, but particularly the organotins, which are used commonly as fungicides. Now, the reason why this is a big problem for the assumption that high-dose testing predicts low-dose results, what would have happened if instead of 1 part per billion, this animal had been exposed to 1,000 parts per billion? You think it would have exploded like that bladder? It would have been thinner. It would have lost weight compared to the control. So high-dose tests predict something very different from what actually happens at really low levels. This is not a surprise for endocrinologists. There are multiple mechanisms by which different things happen at low doses than they happen at high doses. Biphasic dose-response curves are taught in first-year endocrinology. It's what an endocrinologist expects. A non-monotonic dose-response curve is where the sign of the slope of the curve changes at some point from plus to minus or minus to plus, or in a more complicated way over the full range. And as endocrinologists started to look into toxic phenomena, they began to say, wait a second, we're actually seeing non-monotonic dose-response curves, biphasic dose-response curves quite commonly. Often, if you think about what happens in the literature, if you don't have a dose response, the paper's rejected. Some of the papers like this would have been rejected, even though these statistically are quite significant. So a couple years ago, we set out to do an ambitious review in the literature. Anna Soto was one of the authors with us, and we published this in Endocrine Reviews last year. Laura Vandenberg was the lead author, and I encourage all of you to look at this paper because it includes 850 or so references. It's a massive review. It includes data from in vitro experiments, data from lab experiments of animals, and actually human epidemiological data. It looks across a range of types of compounds that include pharmaceuticals, include food contaminants, it includes pesticides, industrial contaminants. Linda Birnbaum, the head of the National Institute of Environmental Health Sciences, when we released this, wrote an editorial about it in Environmental Health Perspectives. The question is no longer whether non-monotonic dose-response curves are real and occur frequently enough to be of concern. These are common phenomena with well-understood mechanisms for the most part. There certainly is uncertainty about some of the mechanisms, but we know a lot about them now. As I said, in our review, we found many examples in vivo and human epi of different types of compounds that demonstrate non-monotonic curves. So let's do a thought experiment. How am I doing on time? Great. Let's do a thought experiment here with one endocrine active compound called Tamoxifen. How many of you know what Tamoxifen is? It's a really important drug for treating breast cancer, and this graph shows why that's the case. That's showing the effect of changing dose of Tamoxifen on the growth of a human breast tumor. The gray line is the control. You can see at high doses, almost parts per thousand levels, Tamoxifen suppresses the growth of the tumor. Now, if you follow this literature at all, you know that one of the issues of interest is what happens when pharmaceuticals get into the water and into drinking water. So if when women take tamoxifen, they do not metabolize it completely. They pee some of it, it gets into the sewage system, and that means it ultimately can find its way back into the water supply. So how will we establish a safe level of exposure for tamoxifen? Let's use these data to figure out what it is. And here you can see the reds. These are experiments from the University of Missouri. We published this in the big review with Vandenberg et al. You can see that there are two statistically significant suppressions of tumor growth. And then you can see that there's an obvious NOEL. Okay? And then you apply, in the US, we apply 3 safety factors, one for animal versus animals, one for children versus adults, one for individual variability. And so that is the safe level using standard procedures. And obviously, you never test that. That's not part of standard regulatory toxicology to test what actually happens at that level. Well, actually, Wade Welshans did test what happens at that level. At the safe dose, the concentrated safe dose, tamoxifen stimulates tumor growth. This is well known. It's called a tamoxifen flare. When a physician is treating a woman with tamoxifen, the goal is to get, as that titer is moving up with treatment, the goal is to get her through that phase as fast as possible because it's painful because her tumor is growing. And that physician makes sure that she stays on that dose regimen because if she falls off it, it drops back down in that dose, at which point the tumor again begins to grow. So what's the safe dose? Well, in this case, let's use the same calculus logic. Let's use those safety factors. There's the true NOEL, according to these data, and there's the safe dose. The message here is we're going to have to find out ways to clean tamoxifen out of the sewage system. And we have some clues about how to do that, but it's not yet being done. So we've got some challenges ahead of us as a result of this. This is a great time to be working in this field because there are all sorts of discoveries because of the new tools of modern molecular genetics and epigenetics that scientists in this issue are using, taking advantage of. I think we need to start using some of this, asking questions about how we are thinking about applying TDCs. Does that make sense in light of some of these weaknesses, or are you all willing to ignore some of the false assumptions that I think clearly are occurring, at least with respect to endocrine disruption? And for me, I spend a lot of time on this now working with chemists, giving them design signals to make safer chemicals and to make money doing it. Thank you.